Okay, ma'am. Start recording. Is the screen visible?
Visible ಇದೆ ಪೃಥ್ವಿ.
Good morning, everyone. I am Prithvi and I am here with my teammates Priyanka and Kirtana of Batch 20. We would like to welcome our guide, Professor Yashaswini AR, and our reviewers, Professor Akshata and Professor Honoraju, on our presentation on sugarcane leaf disease detection and pesticide recommendation system. Let's start with the introduction. Sugarcane is grown widely across the globe and it is a kharif crop that needs hot and humid climate with an average temperature of 27 degrees Celsius. And the ideal soil is deep, rich and loamy soil that it requires. And 180 million tons of sugar is being produced by the industry and independent producers of all sizes. 80% of the sugarcane comes from sugarcane and 20% from the sugar beet. Brazil is being the lead producer with 62% of global output, more than 50% of which its output is being used as fuel ethanol. It is also strongly argued that the production crop severely decreases once it has been affected by the diseases that we see every day. And it has created the urgency to find a mechanism which could put forecast. And now I would like Keetana to continue with the objectives. Keetana?
Yeah, Prithvi. Thank you, Prithvi. I would like to continue the further discussion. Main objectives of the project is to collect and load data set related to sugarcane plants, and to recommend pesticides based on disease, and to classify the images of leaf data set according to the diseases, and to pre-process the collected images and detect the affected areas. Next, move to the paper 1. In this paper, detecting sugarcane diseases through adaptive deep learning models of convolution neural network. There are several methods have been used to counteract problems with plant diseases. and one of these methods is CNN. The CNN method includes a deep learning method that uses artificial neural network. This paper trains different models of CNN to predict the type of sugarcane disease based on feature of leaves and to compare the best model in detecting and recognizing the sugarcane disease. Deep learning engages in developing artificial human intelligence and automated system. With the popularity of deep learning, it is used in several applications in the field of agriculture, like detection of diseases, naming and plant categorization, fruit grading of images, images captured from a camera or attached on robot. Uh, three CNN architecture were analyzed in this study of identify disease from sugarcane plant based on the leaf images. First one is tided net, LeNet, and VGG. Convolutional layer converts the filter of an input image to produce output. An activation layer helps learning a complex image pattern and serves as decision function. In the pooling layer, downsampling to lower neuron size. Fully connected layer identifies very high-level features that highly correlate to an object or class. Let's propose methodology. Image dataset acquitization. Pre-processing of images includes reduction in image size. Feature extraction extracts feature from the resized images. Compile and train the model ratio of 80% used for training set and 20% used of testing set. In the result, the highest validation accuracy of 95.40% obtained over 40 epoch of training using VGGNet model. LeNet model achieves the rate of 93.65% and 90% was achieved by the standard net model. In the conclusion, three deep learning models applied in the study to detect and identify sugarcane leaf diseases. The architecture used in the study compares LeNet, standard net, and VGGNet models. This study can be used as decision tool to assist and provide farmers the familiarity identifying and recognizing the sugarcane diseases that can found in the study. And in the second paper, second paper is portable yellow spot diseases. Identifier on sugarcane leaf via image processing using support vector machine. Involves image processing, support vector machine, yellow spot disease features extraction, normalized difference validation index. The study aims to develop portable yellow spot disease identifier on sugarcane leaf via image processing using support vector machine. The three activities involved are develop a system that captures and displays images of sugarcane leaves and leaves infected by yellow spot using of system support vector machine. Validate the system using samples. The system consists of image characterization, application of support vector machine, data capture and display. In feature extraction, consists of two subprocesses and it gathers data such as energy, contrast, RGB values, etc. Classification process is the brain of whole system. The hardware system of the portable yellow spot disease identifies the on sugarcane leaf by image processing using support vector machine. The research group sampled different number of datasets for each category. 81 dataset for ELF, 99 for yellow spot, and 84 for ring spot. To validate the system, the researcher conducts a controlled and uncontrolled validation by this survey upon completion of the research product. The researchers are able to complete the main objectives of producing a system that can identify yellow spot disease on sugarcane leaf via image processing using support vector machine. Further, will continued by Priyanka. Thank you.
Thank you, Kirtana. The third paper is Sugarcane Disease Recognition Using Deep Learning. The authors are Sammy V. Militante, Bobby D. Giardo, Ruzi B. P. Medina. This study trained and tested a deep learning model consisting of 13,842 sugarcane image dataset of disease-infected leaves and healthy leaves, achieving an accuracy of 95%. The trained model achieved its purpose by detecting and classifying sugarcane images into healthy and unhealthy or diseased class of sugarcane leaves. Therefore, this paper provides an idea of helping farmers with the aid of deep learning algorithm in detecting and classifying sugarcane diseases. In this study, researchers used the convolution neural networks as the basic deep learning method. CNN is one of the most prevailing methods in demonstrating complex methods and uses a large amount of data to perform pattern recognition application. The next paper is Detection of Sugarcane Disease and Classification Using Image Processing. The authors are Arpan Kumar, Anamika Tiwari. This research is carried out to study effectiveness of image processing and computer vision techniques for detection of diseases in sugarcane plants by observing the leaves. Few major diseases in sugarcane plants like red rot, mosaic, and leaf scald have been studied and detection algorithm for the same has been implemented in this research work. During the training phase, various types of leaf images containing diseased leaves are read. Pre-processing is applied to the images to improve the perceptual qualities and improve brightness of the images. Clustering is performed using K-means clustering algorithm. Based on this, the image is segmented into various regions containing disease part and non-disease part. Gray level concurrency matrix algorithm is applied on the image to obtain a database of the features. During the testing phase, the test images are input to the system and the same process of pre-processing, k-mean clustering based segment is performed and the gray level concurrency matrix feature for current images are evaluated. The multi-SVM classifier is then used to classify images into types of diseases. And the next paper will be explained by Prithvi.
Thank you, Priyanka. This paper is about detection of diseases in sugarcane using image processing techniques, and the authors are K. Tilagavati, Kavita, Divya Prabha, Arokia, Joseph Arena, and Sahana. And this study focuses on detecting various sugarcane diseases using digital image processing and developing a web application for the farmers. The system collects the images of the leaves and processes by the means of adaptive histogram equalization. It is nothing but image processing technique that adjusts the context of an image by using its histogram. And then the process of segmentation is done through an adaptive k-means clustering algorithm. The desired features are extracted by GLCM and principal component analysis. Finally, the classification of diseases is done through support vector machine. Support vector machine is nothing but that it collects the mixed data and classified it into several subclasses. Here, the authors create a web application for farmers to access, where it can contain the details of causes and symptoms of the sugarcane diseases. This to serve the function of a watchdog, image process has been developed accuracy of proposed system of to 95%. Coming to the next paper, early identification of leaf stain disease in sugarcane plants using speeded up method robust features. The authors are Romi Fadila Rahmat, Dani Gunawan, Sharfina Faza, Karina Ginting, Erna Budiati Nababan, and they here discuss that their goal is to identify leaf stain in a sugarcane leaf, and by the mean that they speeded up method robust is that it is used for tasks such as object recognition, image registration, and classification. And the input they have used here is the images of sugarcane leaves that has stain spot for image processing using SURF method. The study which they had done in this paper shows 92% of accuracy. Leaf stain is majorly divided into three types, that is ring stain, rust stain, and yellow stain, which is also called like ring spot, etc. As you can see in the image, this is called a ring spot disease, which is caused by a fungus called Epicoccus sorghum and is spread by wind or rain-borne spores of the fungus. It requires less humidity conditions to multiply and generally affect older leaves. And this is yellow leaf disease, as you can see. It is affected by SCYLV. The yellowing extends from the midrib across the leaf blades during monsoon season, and it is easily see through naked eye. Next one is red rot disease. It's a very serious crop infection, appear to be a dull red to brown shades on nodal region that is in the middle part, and this can be treated by using Bavisting, which is an insecticide. Insecticide is a category that comes under one of the pesticide, and it can be used like 0.1% for 15 to 20 minutes. And the next paper will be discussed by Priyanka.
Thank you, Prithvi. The next paper is a novel deep learning framework approach for sugarcane disease detection. The authors are Sakshi Srivatsava, Prince Kumar, Noor Mohd, Anuj Singh, Fatih Singh Gill. A novel deep learning framework approach is proposed in this paper to detect whether a sugarcane plant is diseased or not by analyzing its leaves, stem, colors, etc. The study comprises three scenarios based on the different feature extractors, namely Inception V3, VGG16, and VGG19. These are the pattern models on which different classifiers are trained. State-of-the-art algorithms are compared with deep learning algorithms like neural network, hybrid AdaBoost. Several statistical measures such as accuracy, precision, specificity, and sensitivity are calculated using RN software, and the scenario having the highest accuracy is chosen. A novel deep learning approach was improved in this proposed work. Three different features extraction model, namely VGG16, VGG19, and Inception V3, were used for the extracting features. The output from the feature extraction model was then fed to seven different classifiers, namely Navy Base, AdaBoost, Neural Network, Stochastic Gradient Descent, K nearest neighbor, Support Vector Machine, and Logistic Regression. VJG16 was chosen as the feature extractor model and SVM as the classifier. Hence, more accurate disease detection can be done if more data is available. Thank you. The next paper will be explained by Prithvi.
Thank you, Priyanka. The last paper is about identification of sugarcane. sugarcane foliar diseases and its methods and datasets. The authors are Swapnil Dadabhau Dapal and S.M. Koili. This paper addresses the concern and the types of foliar diseases and researchers undertaken to overcome the problem related to sugarcane leaf disease. Estimated loss is much severe for the sugarcane crop via foliar diseases. They are rust, eye spot, mosaic chlorosis, etc. Use of video signal as input helps the system to predict the dynamic psychological behavior of the plant, and databases with proportionate variety and good example is a key requirement. Noise removal and capturing image is dealt to avoid degradation in performance. Minimum of 2000 images must be database size, so this can be further increased by augmentation methods to avoid overfitting. DNN provides the good choice for identification of the diseases. It helps in enhancing classification accuracy and performance of the system. And this is about the paper 8. Thank you. Ma'am?
Hunraju sir, Akshata ma'am, you can ask questions.
Hello.
Yes, sir.
In the paper, you have not mentioned which type of paper, whether it is IEEE paper or journal papers, and what is the
Yes, sir.
survey finding?
Everything. Yeah, like Lokesh sir had told that only experimental and like Springer, IEEE, from there only you have to choose. So we didn't mention it there.
You could have mentioned in the slide itself. So then, based on that, we can easily understand which type of papers. Then, what is the standard of that paper? So then we can understand whether that idea can be used for your further implementation.
Okay.
So what is the overall finding of your survey?
Sir, they have used VG16 and VG19 and they have compared 2-3 different algorithms and to find a more accurate algorithm. And we are planning to choose which algorithm is the best for our project.
So which dataset they have used?
The data set they have used according to my papers are one that is being infected.
Okay. Infected. Then?
So that it can be easily classified on what type of diseases that is. We've chosen like red spot, dust stain, which is happening in a nearby locality like that.
Whether the
So that,
datasets are standard datasets or just dataset collected by the authors?
I think it's by the authors, as I've mentioned in the papers.
So then where you get a dataset? So
we'll collect it from Google.
You could have mentioned in the slide itself. So these are the survey findings. So then we have a plan to implement this method, and we are going to detect this type of disease. So then that will be very advantageous. Because for the next presentation, we can easily ask questions related to that. Again, in the next presentation also, we will ask about which type of algorithm you're going to use, which data set, which type of disease. So ಈಗ ನೀವು ಈಗಲೇ ಈ ಪ್ರೆಸೆಂಟೇಷನ್ ಮೆನ್ಷನ್ ಮಾಡ್ಬಿಟ್ಟಿದ್ರೆ ನಮ್ಗೆ ಅದು ಈಸಿ ಅನ್ಸಿತ್ತು. ಯಾಕಂದ್ರೆ ನಮಗೂ ಸಜೆಷನ್ ಕೊಡಕ್ಕೆ ಇಲ್ಲ ಇದನ್ನ ಮಾಡ್ಬೋದಿತ್ತು, ಇಲ್ಲ ಇದನ್ನ ಮಾಡಕ್ಕಾಗಲ್ಲ ಹೇಳ್ಬೋದಿತ್ತು. ಸೊ ಅದನ್ನ ಹೈಲೈಟ್ ಮಾಡ್ಬೇಕಿತ್ತು ಅಲ್ವಾ?
ಎಸ್ ಸರ್.
ಓಕೆ, ಇದನ್ನ ನೀವು ಗೈಡ್ ಜೊತೆ ಡಿಸ್ಕಸ್ ಮಾಡ್ಬಿಟ್ಟು ಮೇಡಮ್ಗೆ ಹೇಳಿ, ಇದು ಈ ತರ ಡಿಸೀಸ್ಗೆ ನಾವು ಇಂಪ್ಲಿಮೆಂಟ್ ಮಾಡ್ಬೇಕು ಅನ್ಕೊಂಡಿದೀವಿ, ಏನ್ ಮೆಥಡ್ ಯೂಸ್ ಮಾಡ್ಬೋದು ಮಾಡ್ಬೇಕು ಅನ್ಕೊಂಡಿದ್ರ ಅದನ್ನ ಮೇಡಮ್ಗೆ ಒಂದ್ಸಲ ಇನ್ಫಾರ್ಮ್ ಮಾಡಿ. ಸೊ ಜೊತೆ ಮೇಡಮ್ ಜೊತೆ ಕೂತ್ಕೊಂಡು ಡಿಸ್ಕಸ್ ಮಾಡಿ ಇದನ್ನ ಆಗತ್ತಾ ಇಲ್ವಾ ಅಂತ. ಈಗ ಸೆಲೆಕ್ಟ್ ಮಾಡಿ ಕೊನೆಯಲ್ಲಿ ಡೇಟಾ ಸೆಟ್ ಸಿಗ್ದಂತೆ ಆಗಿಲ್ಲ ಅಂತಂದ್ರೆ ಸುಮ್ಮನೆ ಮೇಲೆ ನೀವೇನೇ ಪ್ರಾಬ್ಲಮ್ ಫೇಸ್ ಮಾಡ್ತೀರಾ. ಮತ್ತೆ ಇನ್ನೊಂದೇಂತಂದ್ರೆ ನೀವು ಅಲ್ಲಿ ಮೆನ್ಷನ್ ಮಾಡುವಾಗ ಸುಗರ್ಕೇನ್ ಕಂಟ್ರಿ ಮೆನ್ಷನ್ ಮಾಡಿದ್ರಿ. ಸೊ ಅಲ್ಲಿ ಡಿಸೀಸ್ನು ಡಿಟೆಕ್ಟ್ ಆಗಿದೆ, ಮತ್ತೆ ಲೋಕಾಲಿಟಿನೂ ತಗೊಂಡಿದ್ದೀರಾ. ಕೆಲವೊಂದು ಪೇಪರ್ ನೋಡಿದ್ರೆ ತಮಿಳ್ನಾಡು ಒಂದು ಪೇಪರ್ ಇತ್ತು. ಸೊ ಕರ್ನಾಟಕದ್ದು ರಿಲೇಟೆಡ್ ಯಾವ್ದಾದ್ರು ಪೇಪರ್ ಇತ್ತಾ ನೀವು ಸರ್ವೆ ಮಾಡಿರೋದ್ರಲ್ಲಿ?
ಸಿಕ್ಕಿತ್ತು, ಬಟ್ ದಟ್ ವಾಸ್ ನಾಟ್ ಇನ್ IEEE ಆರ್ ಸ್ಪ್ರಿಂಜರ್. ಸೊ
ಮತ್ತೆ ಇನ್ನೊಂದೇನಂತಂದ್ರೆ ಕೆಲವೊಂದು ಡಿಸೀಸ್ ಆ ಲೋಕಾಲಿಟಿಗೂ ಡಿಪೆಂಡ್ ಆಗಿರುತ್ತೆ ಅಲ್ವಾ? So first you collect some papers related to Karnataka related diseases. So locality diseases. So then you can forward into your project work. Otherwise, it's very difficult for you to collect your data set.
Yes, yeah.
Okay, sir.
Okay.
Any other questions from Akshata ma'am?
ಅಕ್ಷತಾ ಮ್ಯಾಮ್, ಅದೇ ಸರ್ ಏನೇನೆಲ್ಲ ಕ್ವೆಶನ್ಸ್ ಕೇಳಿದ್ರು ಅದೆಲ್ಲದನ್ನು ನೀವು ಗೈಡ್ ಜೊತೆ ಆಲ್ಮೋಸ್ಟ್ ಎಲ್ಲ ಕ್ವೆಶನ್ ಸರ್ ಕೇಳಿದ್ರಲ್ಲ. ಸೊ ಅದ್ ಕಂಪ್ಲೀಟ್ ಇನ್ಫಾರ್ಮೇಶನ್ ಅನ್ನ ಮ್ಯಾಮ್ ಜೊತೆ ಮಾತಾಡಿ ನಿಮ್ ಗೈಡ್ ಜೊತೆ.
Okay ma'am.
ಸೊ ಅದಾದ್ಮೇಲೆ ನಮ್ಗೂ ಗೊತ್ತಾದ್ರೆ ನೆಕ್ಸ್ಟ್ ನಮ್ಗೂ ಕೇಳಕ್ ಗೊತ್ತಾಗುತ್ತೆ ಅದ್ರ ಮೇಲೆ.
Okay ma'am.
Okay ma'am.
Take that into account.
Okay, Prithvi, we'll wind up now. Thank you, Onraju sir and Akshata ma'am. Prithvi, we can wind up now.
Thank you, sir. Thank you,
you.
ma'am.